Hi everyone, my name is Nadia and I'm the community environmentalist here at T-Town. I have a feeling that a lot of you already know who Leon is, and maybe not so much what T-Town is, but we are a nature preserve and environmental education center located kind of on the cusp of Ossining and Newcastle and Cortland and Yorktown. We have 1,000 acres, 15 miles of hiking trails, and we encourage you to come over and say hello. While I do believe that a lot of you are very familiar with Leon, I will of course let everybody else who might not be aware of his expertise. I'll give a little bit of insight. Leon Chernoff is currently in his 17th year as editor of the internationally distributed magazine Mushroom, the Journal of Wild Mushrooming. In addition, his columns on wild mushrooms have appeared in the Wild Foods Network, Backwoods Home Magazine, and Mycophile, the newsletter of the North American Mycological Association. He is a former president of the Illinois Mycological Association and has given mushroom talks across the country, including, luckily for us, here at T-Town. We're very grateful to have him back sharing his expertise with all of us today. So without further ado, I will hand it over to you, Leon, and if you need anything, let me know. For everybody, please do, if you can, stay muted throughout the conversation so that it kind of reduces the chaos just a little bit, and you can use the chat function and we'll try to get to as many questions as we're able to after. So thank you, Leon, for being here with us.
Great. Thank you, Nadia. Thank you all for coming. So I'm gonna split tonight's talk between some specific mushrooms and some scientific concepts. And yeah, so, and we're entering the truly like late fall period. So I thought I would start out with one of our iconic late fall mushrooms, and which is the bluet. So yeah, and I had this weird thought this afternoon. Oh yeah, I'm going to show you some bluet lookalikes, and with that is a whole bunch of purple mushrooms. So I'm sort of being bipartisan here, but so the bluet is one of our iconic edibles. It gets a lot prettier than this. When they as they get older, they dry out, they get duller. So the ones on the right are sort of fresh and still very purple, and the ones on the left are drying out and becoming a bit tan. And you can see it's usually not round. It's got a general mushroom shape, but the margin is usually, the edge of it is usually tucked under, and it gets this sort of wavy, irregular shape around the edges. And I wanted to show them also, you know, my top priority here is not so much eating, although this is a wonderful edible mushroom, but just showing everything that's here. I remember one time when Alan Bissett gave a talk, and he started out with a slide that showed this beautiful little Baltimore Oriole with all its bright colors, and the caption was "Edible Songbirds of North America." So we tend to look at mushrooms from the standpoint of edibility, but I wanted to look at other mushrooms that you could mistake for this, and as a side bonus, they're all kind of purple and gorgeous. So one of the things about the bluet is it's in a genus broadly construed called Clitosobe. And that is, do we even have a top shot here? Yeah, it's called Clitosobe nuda, which, so it's named for having a smooth top to it. It's just smooth and shiny. And it has white spores. So these gills produce spores that are white. If you leave it on a piece of paper overnight, it'll deposit a white spore print. And the main group of other purple mushrooms in North America is Cortinarius, which has brown spores. So you can see the gills on the bigger mushrooms here are starting to turn brown because they're producing the spores and they're sort of this rusty brown color. And that will color the gills eventually quite deeply brown. And you can see they've got the same general build as the bluet, but these particular ones, we don't know what the species is. They're probably not named. Quartnarius is the largest genus of gilled mushrooms in North America. There are 1,100 species that have been named worldwide, and that's after people staying away from them because they're too confusing and only one person in this country really has an idea of what to do with it. And this particular Cortinarius, it not only has this lovely sort of steely blue shiny color to it, but you can sort of see that the cap is streaked with these darker blue fibers. It's sort of like the cap of a portobello has those little brown hairs on it. This one has these very deep blackish blue streaks of hairs on it. And it's a very distinctive feature, and we don't know of anything that's named that has this. So it's probably unknown. And this happens quite a bit with mushrooms. We really don't have enough mycologists in the country to work with the mushrooms that we have. So there's a tremendous backlog. We actually sent these, a buddy of mine who took this photo, John Dank, we collected them, we sent them to Joe Amirati at the University of Washington. And he said, that's wonderful. I don't know what they are. I'll, put them in my queue. And, so I should get to them in about 8 years. And that was probably around 15 years ago. So I don't know what's going to happen with them. And so we just have this tremendous lack of mycologists in the U.S., and that's getting worse. In fact, in Joe's specific case, the university has let him know that they're not going to hire anyone to fill his position when he retires. So he's just sort of hanging on there, and who knows what'll happen in the future. Anyway, so this is an unknown Cortinarius. Again, we got a couple of new people while I was talking. So we're covering the purple mushrooms that you can find around here. And Cortinarius is what we call a mycorrhizal species. It is symbiotic with the trees around it. The mycelium, the main body of the fungus, which sort of goes through the soil and gets the mushroom its food, is symbiotic with trees. It'll hook onto the roots of the trees around it. The tree will send the mushroom about 1/8 of its sugars during the course of the summer. The mushroom collects water and minerals for the tree. Most plants get about 1/3 of all their water from their fungal partners. And the mycelium, which is the main body of the fungus, it goes through the soil and it's only 1 cell thick. It usually looks like just some very fine roots that will be going through the soil, or if it's something that eats leaves or wood, it'll be going through the wood and digesting that. But it's only 1 cell thick, so it gets a lot tighter coverage of the soil than a tree does on its own. So it can get a lot more of those minerals that the tree needs. Now the bluet, on the other hand, this is it eats the dead leaves mostly, and sort of any any plant waste that's in the soil. So um, it'll be going through the duff and digesting the leaves and sticks and stuff that's in there. So it can kind of grow anywhere. It can, to a certain extent, it can grow on anything. Some people have grown it on coffee grounds, and it'll grow on just general yard waste. So it'll come up in your leaf pile without any trees around. But the Cortinarius will only occur where there are trees for it to hook up with, and it's actually something of an indicator of a healthy forest, in that they have these. It's something that has these nice relationships going on. So this one is not named. This one, let's see if I can. Oh, there's our steely one. And this is that same unknown Quartnarius, and it's younger, so the gills are this really deep blue violet color. And like I said, it has these brown spores, and the genus is named for having a partial veil that covers the gills when it's young. Let me just, yeah. So when it's young, it has these, I don't know if you can see my cursor, but there's this cobwebby veil that covers the gills when it's young. And so here it's still doing that. And this very fragile spiderwebby veil that this genus has is called a cortina, which is Latin for a curtain. And the genus is named for that, Cortinarius. They thought it was a very special feature, and they wanted to name it for that. And the cortina doesn't just help us because we see that there's a cortina there. There are actually other mushrooms that we'll cover tonight, actually, that have a cortina. But I mentioned that the spores are brown, and here the gills are turning brown from the spores, but also you can see the fibers on the right here, they have turned brown because the spores have dropped on them and the spores have colored them. These fibers on the left are still white, but as the cap expands and it breaks away from the cortina, you can see on the edge of the cap here, there are little brown strands and wisps where the cortina has gotten spores dropped on it and it's still attached to the edge of the cap. So if we look back here, there are still some strands strung between the cap and the stem, but also along the stem here, there is this wispy brown area where there are these little fragile spiderwebby fibers that are still attached to the stem, and they've turned this brown from the spores dropped on them, even though the gills are still very violet. And you can see little bits of that over here on the right and down at the bottom here. And so I'm showing you lots of pictures of our local Cortinarius just because I think it's really beautiful, and you won't see it anywhere else. These pictures in here are of a different group of Cortinarius, and there are various names which I won't get into, but just to say that when you have a huge genus like Cortinarius with its 1,100 species, it's often divided up into sections to make organizing it and comprehending it a little bit easier. And these mushrooms are in the section Cereus, where cereus means silk. So they were named for looking like purple silk on top. And I just think that's a wonderful name, and it's very appropriate, and it helps you remember what they look like and, you know, what belongs with what. And I don't know whether this is shiny enough, you know, whether this is silky conceptually, but you know, I don't know whether they're related because Joe hasn't gotten back to us. But here are some more of these purple silky Cortinarius. Again, you can see the wisps of the cobwebby veil hanging down from the edge of the cap. They've been colored brown by the spores. You've got gills that are starting to turn brown in here. Again, they started out purple, they're turning brown. And the young mushroom here, the gills are still completely violet, but you've got this brown wisp of a cobweb here. And then there's this baby mushroom on the lower right, whose gills are still completely covered, and it hasn't started sporulating yet. So the cobweb is still white or pale purple or whatever color it is. Here's a painting of this class of mushroom from Britain in 1847. And it's funny putting these together with the bluet, because some of these have some of those same bluet-like features of that rolled in edge to the cap, the irregular wavy cap, all that sort of thing. But this one is showing, and you can see on the edge of the cap here, there are those streaks, like we had those dark blue streaks on that steely blue Cortinarius that we started out with. But here you can see something else that is used to delineate sections of Cortinarius, which is that the cap is sticky. And most Cortinarius are completely dry, but there's one section where the cap is sticky, and that is called phlegmatium, for phlegm. And it's probably not sticky right now, but one of the pro tips for getting features off the mushrooms that you find is there are leaves stuck to the cap of this mushroom, and there's even this big leaf in the middle. It's plastered to it. So that tells us that at one time the cap was sticky, and since then it's probably dried up a bit, and it might not be sticky right now, but these leaves are stuck to it, which shows us that it was sticky at one time. And if you have a mushroom that you suspect is sticky, or should have been sticky, or was sticky at one time, you can just wet a finger and run it over the cap, and it'll pick up the moisture and get sticky again. So this is a member of the section of Cortinarius that are sticky on top. And there is one other section that is sticky on top and has a sticky stem, and we'll get to them at the end of this folder. So here's a close-up of the veil on the edge of the cap, this cobwebby stuff hanging up off. That's brown where it's been colored by the spores. And you can sort of see just through the bare coloring that there's a matching area on the stem, a band around it, which has turned brown for the same reason. And this just came in yesterday from England. This is a collection of mushrooms, and there's a whole bunch of these bluets up in the top here. And we've got late fall oysters on the left here. There are some other purple mushrooms on the right. These are amethyst lacarias. There are actual chanterelles here on the lower right. I don't know how that happened at the same time as bluets. I've never seen that before. And at the very bottom here in the middle, these are cold weather chanterelles, yellowfoot chanterelles. And, but, you know, here are your bluets. 'Tis the season for them. And now that you've finally gotten some rain, they might be popping out. And again, the bluets, these are not symbiotic with trees. They can kind of come out anywhere. The biggest collection I found in my neighborhood were beneath a hedge that someone planted a hedge like right next to an apartment building. You know, usually they're at the edge of your grassy area, but someone decided to plant a little hedge right along the apartment building and with grass in front of it. And there were years of leaves that had collected at the base of the hedge, and they made this nice mulchy environment. So the bluets just started coming up there, and it was really wonderful. Here's another young Cortinarius, showing that it still has the violet gills. And by the way, let me know, I hope you can still see this, but sometimes we get this question of what is purple and what is violet? And you've got, let me just.
Leon, I hate to interject here for just a moment, but we had one important question is someone wanted to know if you could indicate the general size of these mushrooms as you go through.
Oh, okay. Thank you.
Thank you.
Whoa. These are just a few inches across. You can kind of tell by the leaves that are in the pictures and sometimes by the fingers. Let me see if I can I had these nicely laid out, but then okay. So, whoops. No, I don't want to start this. Sometimes the question comes up, what is purple and what is violet? Because I grew up with the color purple and and then I got into situations with mushrooms where someone was making a distinction between violet and purple. And these two mushrooms right here, these are purple. It's got more red in it. This color down here, these are violet mushrooms where there's more of a blue in the color. So I just wanted to let you know that in case you were wondering. We get confused all the time by some things that, no one ever told us. Yeah. So most of these, again, you can tell the size by the fern, perhaps. The fern is like 2 or 3 inches wide. So these are fairly large bluets, and these are blackberries in the upper right. Yeah. These are some more purple Cortinarius from our area. They're different from the other one. We probably don't know what they are. We don't know what they are. We don't know if they have a name even. But that's fairly normal for, not just for Cortinarius, but for mushrooms in general. Like I said, we don't have enough mycologists. And the general consensus is that we've named 5 to 10% of the mushrooms in North America that actually exist. When, white people from Europe first started coming over here, we gave most of the mushrooms names that were the same. We assumed they were the same as similar looking things that were growing in Europe. And first of all, that turns out not to be true, but also it turns out that they had concepts of what a species is back then that were much too vague to capture the detail and the variety that we have here, or in Europe for that matter. So there are mushrooms that, were given one name here that was a European name. And it turns out that, okay, not only is it not the same as the European one, but we've got 5 or 6 versions of it here, depending on where you are in the country and what the ecosystem is and things like that. So this is an ongoing issue in all branches of mycology. And so this is one that I wanted to especially present because it's sort of the warp drive Cortinarius, where the cap gets these white spots on it that look like you've entered a star field or something like that. And this is Cortinarius Iodides. This is a photo by Tom Bigelow from the New York Club. And this is a good example of what I just said about how we just find out there are more species than we thought. Because for years, this is Cortinarius Iodides. It's our only North American representative of section Mixacium, which has slime on the cap and on the stem. So if you wet your fingers and you try to pick this, your fingers will slide right off the stem. And it's very distinctive. It's very slender for a Cortinarius. You see it's a lot taller and the stem is proportionally a lot thinner than many of the ones we've been looking at so far. And you can also actually see here that the cap is quite slimy. And you can't really tell so much, but the stem is also. But it turns out we have another thing that looks exactly like this in North America, which is now called Iodioides, which means it looks like Iodides, which is an excellent name for it. But the only way to tell is to touch your tongue to the cap, and it turns out the cap slime in Iodioides is bitter, and Iodes is not. So now you can amaze your friends by touching your tongue to the slime of mushrooms and getting scientific results for it. And they'll all be impressed and amazed by you and want to go out in the woods again. So again, we have this is a Cortinarius. It's the main body of it, the mycelium is going to be down in the soil attached to tree roots and helping them grow and receiving nutrition in turn. But we also have bluets at this time of year, and they'll just be growing in leaf duff no matter where it occurs. They don't need trees. And these are very nice, tasty mushrooms. And we will find unknown Cortinarius wherever we find Cortinarius. And again, you've got the brown spores here that are starting to turn the gills brown. And the bluets have white spores, so they do not change the gill colors. And so let me just pause here and ask if we have more questions. I'm looking at the chat here. So the bluets. Oh, I'm glad that you got bluets in New York City because bluets are amazing. The bluets are the only official edible mushroom of the ones I've been showing you. As far as we know, the Cortinarius are edible, but I do actually, let me show you one Cortinarius. Thank you, you are reminding me. So this is the flagship species. This is Violacious, the white violet Cortinarius. And here you've got lots of Cortina. They're showing the silkiness on top. This is, um, archetypal Alba Violacious. This is a site called freenatureimages.eu, and they have some nice mushroom photos, though they are European. And here you can see the gills starting to turn brown from the spores above the Cortina. Alba Violacious is edible. It hardly has any flavor at all. The flagship species for edibility in Quartnarius is Violacious, which is very violet. And it's not smooth like the other ones. This is in its own section also. It's got this rough, you can see it's got on the right, this big one, it's got all these tiny scales on the cap. And this one also does not have any flavor. But it has apparently a very nice silky texture. So if you like mix it in with things that are, I have a friend who makes soup and purees it. So it ends up as this very velvety soup. And it's a nice thing, but apparently, you just don't want to, there's no point in biting into it because you won't taste anything. And this photo shows another trick for finding out spore color, because the mushroom on the left is growing underneath the mushroom in the middle, and the mushroom in the middle has dropped brown spores on the cap of the mushroom on the left. So you've got a little bit of a spore print thing going there. And I have one friend who knows a lot of mushrooms, and he says outright, there are no poisonous purple Cortinarius in North America. And I'm like, that kind of works until it doesn't work. And, we just don't know what is here. We don't know how to tell the ones we know from a lot of the ones that we don't know in North America. So you just really can't go on things like that. Alba Violacious and Violacious seem to be fairly reliable, both in terms of identifying them and knowing that they won't mess you up. But the Pacific Northwest certainly has deadly Cortinarius species, and they're a lot redder than these, but it's not a genius that you really want to mess around with. So there is no guidance that I can give you for going out on your own and just deciding which of the 1,100 Cortinarius species to eat or not. That's any more specific than what I just said. And as far as an app to identify mushrooms, I am actually working on one because I am a programmer. But there isn't really anything good out there right now. Natalie, you are right that there are often very small differences between the species. The apps that are out there right now are just things that show you a bunch of pictures, and you're supposed to match the pictures that look like what you have. But as you maybe saw, here I'm gonna share the screen again. Your books, Gary Linkoff of blessed memory, who wrote the Audubon Guide, said outright, we just really haven't figured out how to come up with a resource, which in his case was, what a book, to identify mushrooms well yet. Because they are living organisms, they grow, as I pointed out in these pictures, they grow and they exhibit different features at different stages of their growth. They change throughout their lives. Different individuals of the same species will have different proportions and things like that because they're growing under different conditions. We haven't had, no one has managed to publish a book that's big enough to have enough pictures like this. It's something that could ideally only be done by an app because, 11,000 species times however many pictures you would need, you're going to be walking around with an encyclopedia. So we haven't really figured out a good way to do it. And right now the apps are just duplicating the methodology of books, which doesn't really work in that there's just too much material to cover, and the apps are still just using one photo per mushroom. And yeah, so yeah, the app said it was this species. And another thing about the whole app and the book thing is you don't start out knowing how to read the mushroom. You don't start out knowing how to pull features off the mushroom that tell you what it is. Like I was here showing you, okay, you've got this wispy veil, it's turning brown. This shows it has brown spores. This helps you identify it. This is not something you grow up, learning from your parents, like this is a blackberry and this is a raspberry. This is not general knowledge in the general community here. The thing also about, is it symbiotic with trees? Do I need to have a tree here for it to be this mushroom? We have we have two, I'm gonna say species of mushrooms in North America. They're both amanitas. They are responsible for 95% of all the mushroom poisoning fatalities. One of them is this big, beautiful, all white thing called the destroying angel in sort of vague, loose terms. And that has been split up into at least half a dozen species at this point. So it's another thing like that, but I'll call it a species complex for the moment. It's an amanita. These two mushrooms are both amanitas. They're symbiotic with trees. And there was one time when I was just getting out of college and I was exploring this new forest, and it had all these mushrooms. It was just really exciting. It was a great forest. And there was a road passing through the middle of it, and you could walk underneath the road. There had been a road underneath. There was a bridge, and you could walk under the bridge, and it was solid concrete under the bridge. And there was this drift of leaves that had piled up about halfway in, and so it's this leaf pile about 10 feet long and 5 feet wide and 3 feet high, and it was covered with these huge pure white mushrooms. And I nearly had a heart attack because it was like, oh no, like the world's greatest concentration of poison that I'd ever seen. But then I was like, okay, wait a minute. This is under the bridge. This is growing on solid concrete. There are no trees attached to this leaf pile. This is not an amanita. It's an all white mushroom, but it's a different kind of mushroom that is feeding on the leaves all by itself. So I checked them out a little more, and they were agaricus. They were basically a wild version of the button mushroom that you get in the store, which is also all white. But these had long, slender stalks. They were the woodland agaricus. And there were features that I could look for. They have chocolate brown spores instead of white spores like the amanita. They have this very lovely almond or anise scent to them. They actually smell sweet. And so I was able to reassure myself gradually and, methodically that these were not things that were going to kill me. And, eventually I learned to do this uh, on a more casual basis, without the heart attack stage that comes when you first think you have something that's like really deadly. So, this is another thing about having an app where people, I think a book is better in that a lot of books do go to the trouble of having discussions of what you might find, what it might look like, what to look for, how to tell things apart. Whereas an app, we're used to just looking at our phones and going next, next. And the apps that I've seen really are set up to just show you a bunch of pictures and you scroll and you swipe, and it's almost like dating or something. But they you have a lot more trouble with that when they don't tell you what features you're looking for, what you should be looking for in your mushrooms. So I want to go quickly. I'll just head right to another family. Oh, so another thing about the bluets is that actually most of the Cortinarius are summer mushrooms. So your probability of not finding them right now is kind of low. The bluet is a real end of the season mushroom. So you actually have a lot better odds of your purple mushroom being a bluet at this time of year. I want to show you one other end of the season mushroom here, or it's actually a group of mushrooms. And that is the genus Hyphaloma. Let me see. So if we go back to the browser, yeah, this, what do I have here? That's the elbow violacious. Okay, so you've got, so this is, you can find 3 different mushrooms that look like this at this time of year. And the edible ones are called Bricktops, and they're a bit redder. They all have this sort of yellowish tan cap that goes a bit deeper colored in the middle. These are black spored. They also have a cortina, and you can see the cortina at the edge here, and the black spores up here at the top have colored the cortina. They've landed on the veil and colored it. As with the Cortinarius, you get this black band on the stem where the cortina, where some of the fibers are remained attached to the stem, and they've been colored by the falling spores. They come up in clusters. These are wood-inhabiting mushrooms. They come up from stumps, dead logs. When they're young like this, the cortina is completely covering the gills. It's just starting to tear and spread out and become a little more cobwebby here. There's actually a nice, this is the set of photos from the Wikipedia article on them, on Bricktops, and which are named for being, a little redder on top. And that cortina, the little wisps, you can see them attached to the edge of the stem here, a little bit here, just as a sort of a weird fringe. And like I said, you get clusters of them from fallen wood. I'm not sure. Again, they're a little bricky. They've got the fringe. Yeah. And a lovely photo. So some of them, this is one of the mushrooms where their clustered growth is one of the features to look for, along with all of these. See, this is lateridium means brick colored, and they. I just want to show you. Hyphaloma. Scientific name for the genus is Hyphaloma. And we have a poisonous Hyphaloma that's called the sulfur tuft, and it basically looks the same, but it's yellowish, more yellowish. And the gills can be a little green. And this is an illustration from a book in 1814, and they did a really good job. Here's a illustration from a German book in the 30s. And again, you can see the relevant features. It's clustered. It's got this yellowish tan cap with a deeply colored center. And this is, again, this yellow one has a common name of the sulfur tuft. So you're talking about brick colored versus sulfur colored, but the sulfur one does get very dully colored, and it looks a lot like the bricktop. And you have to taste it. If it's not, again, the gills are a little yellowish green here. It is safe to taste like a little bit and spit it out again of just about any mushroom. And this one is poisonous. It's also bitter. It has the colored gills. The bricktop, a little redder, it has what we call a mild taste. It just tastes like a mushroom. And these two grow on hardwoods. Whereas this is, you see, this is an old photo. Are you still getting me? Nadia, are you still hearing me? Okay.
Yep, you're still good.
Okay. And we also have one of these yellow ones, which is also a late fall mushroom, that grows on conifer wood, which is, and this is the best edible for me. People tend to go for the bricktop because visual, it's red, it's easy to tell apart from the other ones when it's this red. But this one, it's growing on a conifer stump. This is the most tasty for me. And here is just a whole group of them colonizing this stump and sort of leading you off along the path to mushroom land. And so this is just another little species I thought I would cover. Are we cool if I can just do the ecological stuff, and if that's good with everyone. So I just wanted to cover a little bit of what we call white rot and brown rot, because we've mentioned that these mushrooms, the bluets, the Hyphaloma, sorry, Emily has a question. What is the species on conifers? That is Capnoides, and Hyphaloma capnoides. So let me just share again for a second. So here's the bricktop, Wikipedia, and it also mentioned capnoides. So capnoides. And as you can see, it has the picture over here that's just a little bit less red than the other ones we've been looking at. And yeah, I really like this one. I give my bricktops, they get happy, so it all works. So this is a down, and I think, let me see if I can make this. Okay, well, I'll just leave the pictures small. We don't need big pictures. So when you have, how do I do? Most mushrooms that eat the wood of the trees, they go for what's called the heartwood, which is right in the middle of the tree, which is very good for both parties because then the tree still has a solid outer shell. And a tree like this is only going to get downed by a big storm or something. And yes, it does weaken the tree, but in the meantime, this is how you get animal habitat. All sorts of mammals and birds nest in hollow trees. And the fungus is also turning the tree into soil, which is not a process we think about very much in terms of standing trees. But we do know that when the tree falls down, yes, it needs to decay and become topsoil. And this is an essential process for forest health. And so, in Europe, a lot of their official parks are very manicured. They carry out the dead wood. And as a result, you have things like the Waldsterben, where all of a sudden the whole forest starts dying, and they're like, oh, maybe we did something wrong here. But let me skip ahead a little bit. So, wood. Plants have cell walls, and in the case of trees, if you look at their cells under the microscope, they look something like this, which doesn't help you very much. So we've labeled it. So this is for a tree that would be laying on its side. So these water tubes that are labeled in black and are drawn in black are the tubes that carry the water from the ground up through the tree. And then you have these cells. Now, all cells have a plant wall that's made of cellulose, which you see at the bottom left. So it's this white outer layer in the cells. And plants that are not woody only have cellulose, and you can just, grind them up and make them into paper. And cellulose, as you know from paper, is this fairly soft substance. It's flexible. It works well for regular plants, which herbaceous plants, which just bend in the wind, and they're just they're not trying to be hard and support a huge structure the way trees do. Now, trees are different in that the cell wall has an inner layer, which is drawn in brown here, of lignin. And it used to be thought that the wood decaying fungi either eat the lignin or the cellulose because you tend to have one or the other left behind. So the plant, so if you look at the right half of this picture, the right hand part of the picture shows what we call white rot, where there is only cellulose left of the wood. And on the top right, you have what we call brown rot, where only the lignin is left visibly. There's some of each in either case. And on the left here, you see a composite where in real life, things are not so tidy. So you have brown rot and white rot occurring side by side here. So this is what I was told when I was starting out, and this was the wisdom for a long time. And just this past year, it turns out to be wrong. It turns out that all the fungi everywhere are only eating the cellulose. And this makes sense because cellulose is a polymer of sugars. So So a polymer is a molecule that you get by sticking smaller identical molecules together until you get this long string. So you probably know that sugar is a monomer. It's just one molecule. And then your starches, which are a little bit less digestible, are polymers of sugar. So if you take a sugar and you stick 10 or 20 of them together, you get a starch. And it turns out that if you stick together 120 or 200 of them together at a time, you get cellulose. So cellulose is, and the bigger these molecules get, the more indigestible they get, the more difficult to break down they become. So cellulose is too big for us to eat, just as starches, you have butterflies and other insects that feed on sugars in sap, and starches are already too big for them to break down, which is why a big part of why plants do them. So, certain insects won't be able to eat their food stores. And cellulose is another example of that, where they're just taking it to a new extreme. And fungi are one of the few organisms on Earth that can break down cellulose. So yes, all those horror stories you heard in ancient textbooks of like, if we didn't have the fungi, our forests would just be covered with dead trees and they would just pile up and like awful things would happen. And this actually did happen hundreds of millions of years before the dinosaurs. Before the fungi evolved this ability, the trees did pile up when they died, and they got crushed and submerged and ended up forming our oil and petroleum and coal deposits. And at a certain point, the fungi started eating the dead trees instead, and this no longer happened. But the time period in which that happened, the geologists call it the Carboniferous period, because that's when all our fossil fuel deposits got laid down. But now the fungi digest these. And it turns out they can't eat lignin after all. Lignin is also a polymer, but it's a polymer, I forget what it is, but it's a toxic chemical. It's like an alcohol or a gasoline type compound. So your lignin kind of never gets broken down unless it's white rotted, which I'll get to in a second. So this, when you have a brown rotter, it just very carefully, this turns out to be, the white rot turns out to be the first way that fungi found to attack wood. And they have an enzyme that sort of just blows the lignin apart and it oozes away. The fungi do not eat it. They eat the cellulose, but they don't eat all the cellulose. So you end up with all this white stuff left over. But the fungi are always eating the cellulose. And this takes a lot of energy. It's a sort of brutal process. And they've evolved this sort of peels what's the way it's been told to me is it sort of just peels the lignin apart so they can get at the cellulose, and then they just leave and the lignin zips up again, and they just remove it from between all the cells. So when they brown rot, this is the newer way, you end up with all this brown stuff, and because the cells are intrinsically box shaped, you end up with this very square, the lignin, the brown rot wood is very crumbly. It's still pretty hard, but it's crumbly, and it crumbles in these very square cubical ways. So where they do it very much arborists and foresters actually call it cubicle rot, whereas the white rot is sort of long and stringy, and it's very soft, and cows actually like it. Animals that feed on animals that normally eat grass, if you give them this stuff, they turn out to really like it. And they did experiments in Australia, I think, where they fed the cows white rotted wood, and they really liked it, and they refused to eat hay afterwards. So the farmers had to keep producing the white rot for the cows to eat. And here again is that brown rot, which is very cubical. It's crumbly and square in its formations. Yeah. So the brown rotted trees are usually conifers, and the pines, which are another one of our most recently developed group of plants. So a lot of the soil in the Pacific Northwest is just this pure, crumbly brown lignin, and that's what makes up most of their soil out there. This lignin, when it's powdery, when it's really powdery and fine, this is what you call humus. That's basically what humus is. You've heard of, soil being good in as much as it has a high humus content if you've done any gardening. And this is what it is. It doesn't have any nutritional content for plants. It's just, it's this hollow thing, and it's kind of soft, so it's easier for plant roots to get through. It holds water a lot better than, just crushed rock. And, fungi like to grow in it also for most of those two reasons. So that is that. And that is kind of my presentation for the evening. Do we have any last questions?
I think too, if people want to unmute themselves, it's manageable. You can just ask the question out loud.
Yeah. Okay. I taught you everything clearly.
I had a question, Leon. That deep pilot court you showed us that was kind of steely with the dark kind of scale like fibers on the cap, the stringy fibers. What region was that from?
That is all over North America.
I've never seen anything like it.
Cortinarius violacious.
Oh, I thought you said there was one that wasn't named.
The strange one. The dark
The strange one at the beginning. We've only seen it a few times in the south suburbs of Chicago.
Chicago?
Yeah. We don't
Very
know.
Cool.
Yeah. There's no national Quartnarius network to say, hey, we found something like that in Albuquerque. But yeah, we found it a couple of times there. I haven't been back there in a while. It's a mystery. It's really amazing the undiscovered diversity we have in mushrooms here. I live in Chicago, which has really awful forests. It's the habitat where these Cortinarius was growing are these, quote, forest preserves, unquote, that were all planted by the Works Project Administration during the 40s. So they're not even like long-term natural forests. And they do contain combinations of trees that are found nowhere in nature because they were planted. And so it's sort of not surprising that we also get mushrooms that are found nowhere else in nature, but we have a couple of local amanitas that, Rod has been stumped by. We have this Cortinarius, we have at least this one Cortinarius. And just in my neighborhood, we have a small Calvacia, which is the species, where we have, if some of you know giant puffballs, they're these big things that are the size of soccer balls, and they're smooth, and we have a little Calvacia that's just sort of the size of a potato. and it's pale green inside, which is definitely not a feature found in any other Calvacia. And I took these into the Field Museum, and Pat Leacock said, "Oh yeah, I think we've got some of those over here." And it was a collection from like 20 years ago, that one of their other graduate students had made in the city of Chicago, eating the grass clippings in some Chicago park. And yeah, green interior, no name, reliably discovered there 20 years apart. We have no idea what it is. I don't know if anyone's working on them at all to write it up, but this is something that just happens all the time. Every time people go out looking for mushrooms.
That's one of the things I truly love about it. There's always something that will surprise you, and it's always something that you've never seen before.
Yeah, and that's the people who have the hardest time dealing with it. We found are the birders, because you get people who are used to looking for birds who join the mushroom club, and 98% of all the birds in the world are covered. 100% of the North American birds are covered, and there are only about 900 of them at this point. So if you see something in North America that's undocumented, it's been released from captivity, or it's like blown in on a storm or something, and then all the birders get hysterical and they converge on the area so they get to see this bird that they've never seen before. But they go out with a mushroom club, and every single walk you go on, you find things that are not in the books, and even if you could find in the, even if you had all the books, they might just simply be not named, and some of them get very frustrated with this. Not that the rest of us don't get frustrated now and then also, but yeah.
So I have a question about you just said that it turned green, it was eating grass. Does that, so there are all these wild colors. What is that, because of what they're eating, what they're breaking down? Is it?
No, this was a pale green, sort of like, mint ice cream or something. But and it's eating the dead grass. It's like the bluet, it's living on the plant duff. The puffballs, the big puffballs in the genus Calvacia that are usually like soccer ball size, those are usually in meadows. So presumably they're eating the dead grass and, the dead weeds that grow there. But they don't, all of the puffballs we know, they eat the same stuff, but they're solid white inside. They look kind of like tofu, a big ball of tofu with a rind on it.
And what about the little brown ones?
The little brown mushrooms or little brown puffballs?
No, the puffballs.
There are a variety of things like that. There are some puffballs that'll grow on logs in the forest. There are some that look like that we actually call earth balls, that are actually very hard, and they're actually symbiotic with the trees. They have a completely different lifestyle and genetic affiliation from the puffballs. So once you know things about like the ecology and the relationships, it sort of colors the way you see the organisms. So we've got this group of puffballs that are, whitish and sometimes spiny, and we know that they are eating the plant duff. And then on the other hand, we've got these earth balls that are harder, and they're symbiotic with trees, and they sort of go into two mental categories, even though they look very much alike. So,
okay, thank you.
Sure. Anyone else?
I just have one more question, Leon. I have this strange polypore that showed up on our willow tree in the hollow of a willow tree, and I've never seen anything like it. Can't really figure out any information. I can send you some pictures,
Okay.
but and it basically looked like a Berkeley's polypore, but growing up in the notch of the tree, in the hollow.
Sounds strange.
Shelled, soft, cream white, nice smell. Just really interesting. I don't know anything that associates with willow trees.
Well, I was gonna say Suaviolens, and then you said it looks like Berkeley's, and then so I thought, oh no, but then you said it smells like nice. So, you know, the flagship species on willow is Suaviolens, which means smells nice, and and it usually smells like anise. I mean, it usually doesn't have a stem, and it's usually this little lumpy thing. So it doesn't sound like it looks like what you're talking about, but
It was sort of stemless, actually. It was hard to see, to be honest.
I'll have to send you a picture.
It was inside the hollow of a tree, so we really couldn't check it out that well. But,
okay.
I have some pictures of it in different stages, and I think I have your email address.
Yeah. Cool. Yeah, this is one of the first, you know, when Elias Fries was sort of laying the foundations for mycology, one of his first like exotic collecting trips was up to northern Sweden, and he found that the Sami people in Lapland, they were collecting this polypore, and the guys would stick it into their clothes to make sort of a male fragrance kind of effect and make themselves more attractive. So it's a polypore with history behind it and cultural relevance and stuff.
you could, if you marketed that, you could call it Suaviolens.
Yes, we
Very
could.
suave.
Yes.
Well, thank you very much. We had a great time and learned a lot too.
Great.
Thank you, Leon.
Sure.
We missed Coma this year, so it's great to see you.
Yeah. Anyone else? We okay here?
So much, Leon. I mean, this was fantastic. I hope that we'll see you again soon, hopefully in person.
Yeah.
Because it's so interesting.
Can I ask a quick question? I'm a botanical artist. I teach botanical painting and drawing.
Mm-hmm.
And several of my students have become interested in drawing and illustrating mushrooms.
Mm-hmm.
But we're having, as a matter of fact, two of them are here tonight. And Annie and Gloria is somewhere who's done a couple of beautiful mushroom paintings and drawing.
Mm-hmm.
The difficulty is knowing what we're finding.
Mm-hmm.
And because of botanical art, everything is labeled by genus and species.
Right.
What do you do if it's unidentifiable?
Right. There is notation for this now. You can the Victorian painting I showed you where I was talking about the leaves being stuck to the cap.
Yeah.
I'm putting it in the chat. That was listed as, in the original, as Cortinarius glaucopus, but it almost certainly isn't. But that might be the closest thing that you can easily relate it to. So you use one of these notations to say, you use the AFF in the middle or the CF, and that stands for affinus or confer. And one of them means this is as close as I can get, and one of them means I really don't know what this is, but this is, you know, I'm just going to use this name. And mycologists use these two abbreviations in absolutely inconsistently among them. So, you can, which means that people like your illustrators can use them any way they want. And you'll be on a solid scientific footing as far as current practice is concerned. And, when you're illustrating a mushroom, so as you could probably see tonight, you want to get the underside. If you're doing just a pretty picture, just arrange them nicely and, make a pretty picture. But if you want to show the diagnostic features that show you what it is, you've got to turn it over. You hopefully show it in different ages.
Okay.
And that's, like you were here for at the beginning when I was talking about these wispy veils and colored by the spores and things like that. So you want to show the top and the underside. You want to turn some of them over.
How important are longitudinal cross sections, dissections?
They are mostly important to show color changes because there are lots of mushrooms that will turn color when you cut them, the way like an apple will turn brown. And this can be important in identifying them.
Okay.
The other thing you can show that way is the way the gills attach to the stem or do not attach to the stem. Because there are some mushrooms where the gill, if you take a portobello or a button mushroom from the store and you do a section from top to bottom, you will notice, now that I've told you about it, that the gills do not reach the stem. And when you have a mushroom that does that, all of a sudden that puts it in a completely different group from everything that's attached. So there are a very specific subgroup of gilled mushrooms where the gills are free. They don't reach the stem. There are mushrooms where the gills are notched, where they're going sort of straight into the stem, and then they just do a little dip right before they get there. There are mushrooms where the gills run down the stem, and those again are very distinctive and have their own little group. So it's often easiest to illustrate that by cutting them down the middle. So you can do all these things and it won't hurt. If if it's time sensitive, like your mushroom is drying out, you might not have time to do all the different views that you want to do. But, you can always do, you can lie with painting, right? You can do a painting of the whole thing and then chop it in half and do another painting and put it next to it like they're part of the same painting and stuff. So it's, so those are some things.
Okay. Thank you very much.
Color changes and gill attachment. So.
Leon.
Is that Gloria?
Hello. Leon, my name is Fred. I'm sorry I came very late into this meeting. I did. But I'm going to ask my friend Alan all the questions. We're going to grow mushrooms, but interesting thing is a couple of years ago, I had relatives come to visit me and I never even looked around my backyard. He went out and picked up a bunch of mushrooms. They were from Slovakia. And that's what they do all summer. They go and they do mushroom hunting. And he picked mushrooms. I don't know whether I can show you this, but he actually, I don't know whether you can see that. It was
okay. It looks like a bolete.
Pretty solid.
It looks like you've got a pine tree in your backyard.
Pine trees, oak trees. He found about a half a dozen of these. Yeah. They were pretty big, and I had never seen them before.
Yeah.
It was the best mushroom I ever ate in my life.
Great.
My wife wouldn't eat them. She said he picked them in the wild. I don't know what they are, but
yeah,
fantastic.
Yeah, the first wild mushroom I ever collected was something called a hen of the woods, which Emily was lamenting not having found any this season. But I went out looking for something else. I found a hen of the woods. It's this brown, feathery kind of thing. It looks sort of like a feathery brown cauliflower. And it was 13 pounds.
Wow.
And I cut it up into fifths, and I brought a fifth of it home with me over Columbus Day weekend, visiting my parents. And it was in one of those big, like 20-gallon buckets that you usually store laundry detergent in.
Okay.
And I came home, and I displayed my bucket and my mushroom in it, and my parents went, "What is that thing?" And I said, "It's a mushroom. I'm gonna make it into soup." And they both went, "What?" And my father said, "How do you know that that's edible?" And I said, "Well, I've already been eating the other pieces of this for the past 2 weeks, so I'm here." And my father said, "Oh, well, then it's okay." And my mother went, "What?" No, but yeah, I got them started, and they've been at it ever since. So
Right.
Fred, this was recorded, so I'll be able to send that to you so you can watch the rest of this at a later time.
Thank you. Thank you.
Great. Yeah. Okay, so I guess we've.
All right. Well, thanks again, Leon, and thanks to everyone who took their evening to come hang out with us.
For you all coming out.
Thank you.
Thank you.
Take care.
Have a wonderful evening.
Take care, everyone. Stay safe.
Thank you.
Thank you.
Many mushrooms, everyone.
Yes.
Bye-bye.
Oyster season.
Thank you, Leon.
Thank you.
Thank you, Leon.
Thanks.